Hello, I'm Tim Wall, Senior Reporter for Pet Food Industry. Thank you all for joining us for this edition of Ask the Pet Food Pro, Yeast-Derived Health Solutions for Dogs and Cats. We have a global audience signed up for this chat from Angola to Vietnam, and I'd like to welcome you all. Ask the Pet Food Pro connects experts with industry professionals and offers the opportunity to pose questions directly to those experts. Ask the Pet Food Pro is brought to you by Pet Food Forum and Pet Food Industry's journalists and editors. Drawing on our combined decades of experience covering the dog, cat, and other pet food markets, you can find our reporting and analysis at petfoodindustry.com. Don't forget to subscribe to our daily newsletter and monthly magazine. Today's discussion focuses on biotics as pet food ingredients and how they can be created from yeasts. Biotics producer Phileo sponsors this edition of Ask the Pet Food Pro, and our expert guests are Jan Cukuldoski, Purina Pet Care Endowed Chair for Microbiome Research and Associate Director and Head of Microbiome Sciences at the Gastrointestinal Laboratory at Texas A&M University, and Russ Kelly, Global Program Manager for Phileo by Le Safre. Russ has an extensive history in pet food with 28 years of R&D experience focused on nutritional physiology. Welcome, Jan and Russ, and thank you for joining us today.
Hello.
Thank you. Thank you, Tim. It's a pleasure to be here.
Thank you for being here. So before we start the discussion, I do have a few announcements. Audience members, you've all been muted to ensure that everyone can hear and focus on the discussion. I've included many of the questions you all sent in before the broadcast in our script, but if you have questions that come up during the discussion, please submit those and other comments in the chat box. Please note that this chat is being recorded, so you can access the recording starting in about a week at petfoodforumevents.com. Click on the Ask the Pet Food Pro tab at the top. All right, Jan, let's start with a brief biography of biotics. What are biotics in general?
Well, I guess biotics in general is kind of ways to somehow modulate the intestinal microbiome, right? I mean, the intestinal microbiome is probably the one that we most focus upon, but generally any microbial system, right? And there are different kinds of biotics. I think we evolved over the last few decades, understanding much about the microbiome and how it can modulate them. And I think, Russ is kind of the experts to kind of, from a definition point of view, to kind of address all those different kinds of biotics that are now on the market. And I can afterwards chime in about the health benefits and how it really ties into the GI health.
Good deal. So Russ, I know, like Jan was saying, there are different types of biotics: probiotics, prebiotics, synbiotics, and postbiotics. So let's discuss each type a little bit and how they differ from each other. So first off, what is a probiotic and how do they differ from prebiotics, both biologically and in their functions?
All right. So in terms of that, I'm going to say historically, there's been some confusion around in terms of the use of these various biotics, and the International Scientific Association for Probiotics and Prebiotics. They've done a great job of defining some clarity by putting some definitive definitions around with prebiotics. They define as a substrate that is selectively utilized by the microorganisms that confer a health benefit to the host. And the key there is that for the prebiotic is they are utilized by the microorganism. So the microorganism is in fact their target, whereas the probiotic is defined as a live microorganism that, when supplied in adequate amounts, confers a health benefit directly to the host. So the two key terms there are live microorganism. So a probiotic has to be a live microorganism, and that it has a documented health benefit to the host. And then, of course, we get into synbiotics, and that is I'm going to say, a complex of a pre and a probiotic in combination. And then we have postbiotics, which are defined by the International Scientific Association for Pre and Postbiotics as basically an inanimate microorganism. So it's not a live microorganism, but it is a complex of materials that is provided by that complex that again confers a health benefit to the host. So regardless of whether we're talking pre or postbiotics or anywhere in between, the big key is that the commonality is that they deliver a health benefit to the host.
Gotcha. So to kind of to sum that up, probiotic is something that's going to actually the actual living organism. The
It has
prebiotic,
to be it has to be a live organism, yes.
the symbiotic is a mix of the two, and the postbiotic is the no longer with us, the deceased.
Right. It is also a similar to the symbiotic. It is a complex material, so we can't take like purified metabolites from an organism, and that's not classified as a postbiotic. I'm going to say within the marketing world, sometimes it comes pretty close to people classifying purified substrates or metabolites as postbiotics. But I guess in its clear definition, it has to be more of a complex material, not a purified metabolite.
Gotcha. So considering all those differences, when should pet food formulators use a prebiotic versus a probiotic versus a postbiotic, or put them together to make a symbiotic? What are some of the best uses for each?
Yeah, so I'll start and then I'll let Jan build in terms of that. Um, I think that probably the most common use of the biotics is clearly a prebiotic when it comes to complete and balanced foods. We see this in almost across the board with regard to the at least foods in North America. Um, FOS or fructoooligosaccharide is one of the most common prebiotics that we see show up on labels. And in terms of healthy animals that owners are looking to, I'm going to say just promote health in, certainly prebiotics are a good place to start. And then as we move into animals that are undergoing challenges, we start to see more of the probiotics and the postbiotic utilization. And then of course we may have formulations that use the symbiotic approach where they've included a prebiotic as well as a probiotic um, utilization.
Gotcha.
I'll let you add on top of that.
Yeah, maybe just one step back, actually. I would like extend a little bit one more, not definition, maybe to you, Russ, a question about fiber, right? Because I think there's a lot of confusion between prebiotics and fibers, and I think we hear a lot of fiber-rich diets and so on. And so maybe could you just maybe answer or define, is a fiber prebiotic? Can a prebiotic be a fiber and so on? Because I think when we afterwards go to health benefits, sometimes when you read publications and there's always some people use fiber, some use prebiotics. I think
Right.
it would be important.
Yeah, I think in terms of that, certainly the distinction there is is the fiber being provided? Is it one that is utilized as a nutrient source by the microorganisms? In terms of that, we have you know beet pulp is a common fiber that is utilized, and it has prebiotic uh, characteristics. Whether it would truly be viewed as a prebiotic would be up for debate depending upon who's asking. But certainly, it is a fiber source that, at least within components, is utilized by the microorganisms in terms of their metabolic activity. So with that, it has prebiotic characterizations, but it also has other components that would not fit the classical prebiotic component. But yeah, certainly there are fibers that would fall into prebiotic uh, characterization, and then fibers that would not.
Yeah,
Gotcha.
and so maybe the reason I was kind of asking about going to the health benefits, right? Because, I do this now for roughly, well, I want to say how many years, but almost 25 years, right? And Russ actually and I go back way long, right? When we started with working with probiotics and so on. And I think over the last 20 years, I think we learned so much more about how the whole microbiome works, how it interacts, right? And so in the past, we kind of focused on either probiotic alone, right? Or either prebiotic alone. Then some people focus just on fibers and so on. But I think the more we learn, it makes it very clear that it has to be a combination approach in many cases, right? So ideally, you have to, like gardening the microbiome, it's not a single solution. The combination approach is probably a good one, and it really depends on the stage of the animal, the health of the animal, and so on. So I think that is very important to understand that each of those mechanisms going to address a little bit different areas, right? And it kind of makes sense that potentially synergistic effects or gardening from multiple sides is probably the way to go in the future, right? So that'd be my overall kind of statement in the beginning.
To kind of delve into that a little bit more on the health side of it, we often hear about probiotics, all the biotics, in terms of their effects on GI health.
Yeah.
So what are some means to assess the effects of biotics on gastrointestinal health in pets?
Yeah, and I think that is clear definition, and probably a lot of people are going to have different opinions. What is a health benefit, right? And I think the health benefit ranges all the way from we can see clinical resolutions, right? So yeah, I think in the beginning, the major focus was, can I show with a probiotic, for example, that we can reduce, let's see, dogs with diarrhea, for example, right? If I give a probiotic and diarrhea stops, that is a health benefit, right? Then there's other studies that shown that actually when I give probiotics, I can target immune system. Okay, so I can have clear markers of immune function. Let's say, secretory IgA, which is a marker of mucosal immunity, or markers of inflammation going down. So different strains probably address different solution, right? Prebiotics or fibers, when I really modulate the resident microbiota, it's very crucial to understand that most bacteria in the gut are very strict anaerobes. Okay, so I think that's also a very important feature, that it's very difficult to create a probiotic that is actually an anaerobe, right? So actually, the component where I give nutrients for the microbiome, for those bacteria that are much higher abundance and they're strict anaerobes, that if I give them a fiber source, right, of something or like a prebiotic foods, and I can selectively increase their numbers, it's very attractive, because I don't really have to culture bacterium. It's very difficult to, to create probiotics. So that's one thing. But in the end, I think it's important to understand that each of those components does a little bit different thing, right? And then when you add the thought about fiber, fiber can be a prebiotic, right? But sometimes you have also these insoluble fibers, meaning those fibers that actually just provide bulk for the GI tract, motility, and so on. So I think for me, it's very clear that we learned over the last 20 years, it's not just putting everything into one basket, it's this combination approach, and that probably, addresses digestion and the health of the GI tract from different levels.
Gotcha. Before the show, we had talked a little bit about the dysbiosis index. Could you tell us a little bit about that?
Yeah, so I think that's an important question too. That's we learned a lot, obviously there's a lot of research done in looking at the microbiome itself, right? What is normal, which bacteria are normal and so on. And over time, we just simply were able to identify some of the key bacteria, right? So you think about it, if you have an ecosystem, we can try to target every single bacterium, but often if you focus on some key members of an ecosystem, let's put in a regular ecosystem like the trees, right? If I know the trees are there, that's pretty much important members of the ecosystem, right? If the trees are for suddenly gone, I know I have a severe change in the whole ecosystem and obviously function, right? So what it means that over time and that's really a work of over almost 20 years, right? We start with sequencing, identified key bacteria with repeated studies. We identified that some bacteria are usually always increased in GI disease or some, dysfunction, and those are classical ones like E. coli, for example. And often bacteria that are new, like Faecalibacterium, like, Clostridium hydrononis, those are very strict anaerobe bacteria that we only identified using molecular tools that typically go down in GI disease, okay, or some dysfunctions and so on. And so over time, we discovered that we can actually target those bacteria, do some mathematical modeling, and we can kind of predict how strongly the microbiome shifts, okay? And the dysbiosis index is really a marker for severity of microbial shifts. And that means it's usually really increased only if I have either antibiotic usage, right? Or mostly some chronic GI disease. And we discover now that once you have chronic GI disease, it's a complete, usually persistent chronic dysfunction of the GI tract. So think about it, the soil is going is not working anymore. You have the receptors, transporters, the mucosa, has fibrosis, has less of the transporters, absorptions, and so on. So we have kind of a little bit of malabsorption in the GI tract, and that really drives dysbiosis. Okay. But this is really a severe shift of the microbiome. And we discovered now that actually once you see this severe shift, it's usually a bad sign. It's actually a really bad sign of chronic GI disease. And we have a huge flip in the gut microbiome. And those animals, it's very difficult to actually bring ever back, right? Because it's not just the microbiome. The microbiome reacts to the soil, if you want to see that, right? And the soil is chronically abnormal. And this really requires chronic treatment. And that's where actually this multimodal approach comes into play. It's impossible to fix this with one probiotic or with one prebiotic, right? There we talk about nutrition, probiotics, and so on. So it becomes this multimodal approach to kind of manage this system without chance of cure. So, but in a normal dysbiosis index, it would indicate that we don't have severe shifts in the microbiome. And that's where I can actually probably modulate the microbiome easier. Kind of makes sense, right? A healthier system is easier to modulate than a system is completely broken for other reasons and so on.
Gotcha. So to kind of expand on the metaphor you're using of trees in a forest, there's a big difference between an old growth forest ecosystem functioning versus, secondary or tertiary growth. Could you expand on that idea of how biotics can serve as health interventions to promote health versus disease treatment?
It's exactly the principle, right? Think about biotics as you have a system that kind of is healthy, right? More or less. And you can say there's probably a huge range of health too, right? As you said, a system is still functioning when it's highly diverse, like in a jungle. And yet it's still to some degree going to function a little bit, when it's less diverse, right? But ideally, we want to have this diverse system. And that's what really the biotics do, right? And already in more or less what a GI tract functions normally, adding solutions like a probiotic, so I can increase the diversity. Some of the probiotics, what they can do is then can help with digestion, right? They can break down substances, and those substances again become available for other bacteria to grow. Okay. Some probiotics can reduce a little bit the pH. That means it reduces some of the bad bacteria, and we can give the chances. When you now add prebiotics to that, you already selectively, hopefully that's what the definition is, that I selectively already increase the food for those bacteria I want to grow on them and so on. So again, gardening is a difficult job. I don't know for all of those who garden, it's not something you can do with one miracle, planting something. You have to work from different levels. And that's why biotics is one important component of that, together with a healthy diet, digestible diet and so on. I think that is really where I see the future going, thinking from all sides of that. And so for me, it makes perfect sense that all those biotics should be part of feeding, and which form, whatever, either in the foods or as supplements or something like that. Obviously, that depends on the technical challenges in some of those, how to formulations, right?
And so expanding on the gardening metaphor once again, once there's been something like a clear cut, I know, times when I've had to take antibiotics and things like that, doctors have recommended then using, probiotics and things like that to help you recover from that. So what effect do antibiotics really have on the microbiome, and how can biotics for pets be used to address that?
Yeah, so antibiotics, it depends on the form of the antibiotics, but most of them, and especially those ones we use usually for diarrhea in dogs and cats, those broad spectrum have a really devastating effect on the microbiome. So we have now many, many studies where it's almost like, think about it, metronidazole or tylosin, those are very commonly used antibiotics. They pretty much burn down your forest. So imagine really you drive to an area where the whole forest is burned down, okay? And so, but what happens, and this is not just because the bacteria are gone, the bacteria have important functions, okay? So we suddenly, all the metabolites that are produced by bacteria are not there anymore, right? So really see that way. The good news to some degree with antibiotics is that if the forest is burned down, usually the soil in many cases is still kind of okay, right? So actually many animals, the microbiome grows back, okay? But it's very individual. It takes some time. And in some animals, some key bacteria never recover, okay? But generally, the trend is, it's a very, very robust system. Generally, the system wants to grow back. It makes perfect sense, right? But as I said before, what we want to do is, can we kind of accelerate the recovery? That's number one, right? can we maybe supplement some of those compounds that are missing? This period where the microbiome is not there, it's obviously a vulnerable system. Pathogen can overgrow and things like that and so on. So, that's probably a huge area of interest, right? When you give a probiotic at the same time with antibiotics, that is a really big challenge because, first of all, many bacterial probiotics are susceptible to antibiotics, right? So you cannot give them at the same time. Number two is, I can tell you from our studies, you cannot prevent the dysbiosis that you're inducing, but probiotics can help a little bit speeding up recovery and can also ameliorate a little bit the clinical signs that you have. So antibiotic-associated GI signs, that means a lot of animals, dogs and cats, they're going to have diarrhea like humans have. A lot of humans, you probably know, have complained, oh, I received antibiotics, now I feel this pain and things like that, or I have diarrhea. Same thing happens in dogs and cats. So my point of that is, it's all of this combination, how can we avoid or, ameliorate those consequences? And that's actually where also from a probiotic standpoint, we can actually think about other better options like yeast, for example. Yeast probiotics are those ones that are resistant to antibiotics. In this case, it's a good thing, right? Because we actually give them at the same time when we give antibiotics and so on. So my point of that is, so learning now all those little intricacies can really help us to choose much better, which biotics would be the useful to give, right?
Gotcha. So this is actually following up on that. I've heard of something called fecal microbiota transplants. Could you tell us a little bit about that and the role that biotics play in improving the effectiveness of that?
So yeah, that's a big area, and I think fecal microtransplantation is pretty much the biotic treatment, right? Because you're really taking a fecal sample from a healthy animal. It has tons of bacteria. Don't forget, there's 100 trillion bacteria in the gut, right? So when you take a few grams of a fecal sample, you are giving a lot of bacteria, which is good, right? Then also not that, it has also all those metabolites the bacteria produce. So short chain fatty acids, secondary bile acid. There's so many compounds that are important too. And it's a new area, but we clearly see benefits. We can clearly see that we can use this, but I can also tell you right now, and it comes back to the dysbiosis indexes, once you have severe GI disease, severe means doesn't necessarily be clinically visible, but those severe dysfunction of the GI tract, right? Even FMT can temporarily improve your gut microbiome, but often they relapse because again, the gut function doesn't work, right? So where FMT is really good is in antibiotic-induced dysbiosis because the soil is still working, right? So I can just quickly replenish it. So we have this data. But it comes in these chronic GI disease cases, right? Then again, FMT is great, but I cannot give it every day. So I still gonna have to combine this treatment. I give it every few weeks. But in between, I still need to work on the biome, right? With probiotics and things like that. So I think this is the great exciting area of research that we have to combine treatments, right? And so kind of help it and so on. And we actually see that animals they have diarrhea, but with very mild dysbiosis, the dysbiosis index is actually in the normal range, they respond the best to FMT, right? That's kind of counterintuitive in the beginning, but when you start understanding GI pathology, it makes sense because the gut is functioning. So I can give them one shot of FMT and they're really good for a while, right? But having said that, those animals are going to respond maybe to some probiotics or fiber too, right? Or prebiotics. So I think that's where we're learning more in which situation. And maybe I need to really highlight, I think we discovered much more that there is clear differences between a healthy gut, even with diarrhea, or a gut that has diarrhea but has severe dysbiosis. Okay, that's important, I think. Those are two different treatment approaches there.
So speaking of another illness or concern, before the show, an audience member had submitted a question about the ability of biotics to address leaky gut. Could you discuss that a little bit?
No, I think the leaky gut is a very valuable it's a clear pathophysiology, right? The problem is a little bit ill-defined, right? What does leaky gut truly mean, right? So it's actually very difficult to measure it, and that's a little bit where we have hard time. And what is the best assay? What is the best method, right, to truly confirm it's a leaky gut? Okay, but when you start thinking about the physiology of the GI tract, right, you have to have a very diverse microbiome. That is the very first layer of a healthy gut. then you have this very important mucus layer. This mucus layer really separates all this bacteria from the epithelial cells. The metabolites and nutrients can diffuse to this mucus layer. And then you have the epithelial cells who have to be very tight, balanced, not balanced, and so on. And over time, I think, and that becomes to age or often very simple damage model. I think, often GI disease, chronic GI disease starts with simple damages, right? Think about it. The gut is exposed to a lot of damage, gastric acid, high fat, stress, there's so many different things. And then other factors that we don't know we understand yet, slowly, think about your skin, right? I mean, how much beating does your skin get? Over time, you get the scrapes and so on. Same thing happens in the gut. And over time, all of this happens. And in the end, multiple things happen at the same time, right? You change the microbiome, maybe you don't have this perfect metabolites anymore, right? That is a little low-level pro-inflammatory signaling, right? That ultimately causes inflammation on the guts. That ultimately degrades the mucus layer. Inflammation causes mucosal remodeling. Epithelial cells, they kind of atrophy, and then you have this leakage, right? All my point is this, it's not one simple, you cannot just say it's leaky gut, right? So leaky gut is a combination factor of many different factors, if that makes sense. And again, this is a huge area where we can say, can we have solutions like probiotics that are specifically, improving barrier function, right? All of this stuff is kind of this where we really, really want to show that. And I think we have some data that some of those biotics can really improve this barrier function, right?
Gotcha. So to change, I'm sorry, go ahead, Russ.
No, I was just going to point out, I think Jan had some excellent points in terms when we think about, just the challenges that our pets have. I know that at Pet Food Forum this year, Jan is going to give a lecture on some data that he was part of in terms of an abrupt diet change in terms of its impact and how a probiotic may help, or our yeast probiotic helps sort of attenuate those challenges of an abrupt diet change. We also have Jan mentioned in terms of the impact of the antibiotics. We're going to have a poster at the Pet Food Forum where we've demonstrated that the yeast probiotic Actisaf SC50 that can help attenuate or mitigate some of the abrupt changes that is associated with giving an antibiotic to an animal. So those are exciting things that we're going to be sharing in the future. But I think he also made a really good point that when we start talking about trying to influence an ecosystem, that it's not going to be a simple solution, right? It's going to be looking at various and combinations of solutions.
Yeah, and I think just to emphasize what Ross just said, I think for us, we're going in this direction of prevention, right? The more we can ameliorate all those little insults that the gut receives over time, right? From dietary change, from antibiotics, from stress, and so on. This is really, I think, the most exciting area. And that's why, biotics, especially targeted for health and prevention, long term, probably going to have a better chance because once your system is broken, it's very difficult to cure it, to fix it, right? So I think that's where really biotics shine. The focus is on not stopping disease, but really making the guts function, smoother, if you want to say, and prevent all those little damages.
Gotcha. So we've talked a lot about the importance of biotics in gastrointestinal health, but what are some of the other physiological systems that biotics can support in pets?
Well, I think this is an area that is still a little bit underdeveloped, right? I think the gut is probably the ones we know most because obviously you give it orally, we understand all the bacteria and so on. But long term, I think it's actually maybe even going to be easier to modulate some of those other organ systems easier because there's actually less bacteria, right? Think about the skin, right? In the gut, you have trillions of bacteria. So you have to it's very difficult to give a lot of dose of probiotics, for example. But on the skin, for example, we know there are skin clear, also relationship between the microbiome on the skin and the immune system, or even the barrier, especially and so on. So, skin lotions, for example, we can actually apply a probiotic more long term. Think about it, right? In the gut, it goes through, right? But on the skin, you put a lotion and so on. So actually, you can have much more contact time and so on. I think all those areas, I know that they work on the human sides. I'm not really aware much at the moment on the veterinary side, but it's for me an area that makes perfect sense, right? Or maybe ear infection, where you can say, I'm giving good bacteria to kind of push out all, some of these pathogens and so on. So the applications are potentially huge. It just has not been explored that yet.
Gotcha. Well, now what about the other end of the gastrointestinal system? We had a question from an audience member before the show about biotic additives that can address dental issues. Is there any research on that?
I don't know, Russ, I mean, I know there are some. I don't know if Russ, you maybe know a little bit more specific, and I can maybe do more general ideas,
Right.
but are you aware of some specific applications?
Yeah, certainly that is an area of interest for us as well as others. We Jan participated in a symposia that we had at the animal science meetings in Calgary this past summer, and there we had a lecture on the postbiotic effect on oral health by Dr. Oliveira from Portugal, and it was a great session. We had a really good turnout. We had I'm going to say there was some travel challenges associated with that meeting with some airline disruptions. But overall, it was a really wonderful session, and Jan was part of that as well as Dr. Kelly Swanson, uh, Ananda Felix from Brazil. So we had a really good session in terms of the use of biotics, and oral health was one of those areas that were tackled. Immunology as well.
Immunology as well. Gotcha.
Yeah. I mean, maybe just a general thought about, I mean, we know there's clear data that's pathogenic bacteria in the mouth, right? So if you have gingivitis, if you have stomatitis and so on, you're definitely also going to have a dysbiosis. And some of those bacteria has been shown in some animal models, they translocate to the gut and cause inflammation there. So it makes perfect sense to address, the oral cavity also, with different approaches. But again, it's also probably going to be a multiple approach, right? And I think one technical challenge in the dental space is the contact time. How do you actually keep a probiotic there for long, a biotic there for a long time? And so I think that's probably a technical challenge.
So let's get into a little bit more of the technical aspects of this and kind of explore what the biotics that come from yeast are specifically, as opposed to like bacterial ones. So, Russ, if you could tell us a little bit about how biotics are made from yeast and what we're talking about there.
So I'm going to say in terms of general from a probiotic standpoint, whether we're talking any microorganism, whether we're talking bacteria or a yeast derived, again, it's going to be I'm going to say vary from strain to strain in terms of its effectiveness. So so with that, there's going to be I'm going to say selection that takes place, really looking for unique characteristics for that particular strain of microorganism. Within that, certainly it has to be one that can survive passage through the gastrointestinal tract and arrive into the lower GI tract as a live microorganism, as required to be a probiotic. In terms of our yeast probiotic Actisaf SE50, we have done I'm going to say very detailed characterization to demonstrate that it is capable of surviving passage through the gastrointestinal tract. It does deliver a host of health benefits such as reduced pH in the lower GI tract. It influences the microbiome population, specifically decreasing some of the pathogenic bacteria that are present. It, as Jan mentioned early on, it produces metabolites that can then go on and produce other health benefits and influence the microbiome, both from I'm going to say promoting additional health benefits for the other beneficial microbes that are there, as well as demonstrating some immune benefits. When we start talking about yeast, they are like any microorganism. It's a complex. They're not just producing one metabolite. It is an array of metabolites that they are producing, and they do have. I'm going to say there are some advantages. If we look at like probiotics in general, all of those that are defined as a probiotic are going to deliver a health benefit, whether that be a microorganism or I'm sorry, a bacteria or a yeast in terms of the microbe. We feel like that yeast probiotics has advantages over some bacteria in the fact that they tend to be a little more I'm going to say resistant in terms of harsh environments, so they're able to survive in harsh environments as they transient the gastrointestinal tract. They don't tend to colonize, as we talked about early on. They're not sensitive to antibiotics, and they don't contain antibiotic resistant genes. It's just in terms of their I'm going to say their DNA, right? They're just not sensitive to that through some of the um, pump systems that they have. So in terms of that, there are certainly effective microbacterial probiotics out on the market. We've certainly embrace our probiotic in terms of a yeast probiotic and feel that it has certainly advantages over in a general to bacteria.
Mhm. And I think also yeast is easier to add to the diet, correct? The stability and so on.
Yeah, we've done a lot of work in terms of making sure that our probiotic is stable. Now it's not extrude. I'm going to say it can't go through an extruder, but it is an extremely stable probiotic once it's applied to an extruded food after drying. So in terms of that, it can be post extrusion applied, and it's very stable in terms of that product. We get anywhere from 15 to 20 plus months of stability in terms of those dry foods.
Mhm. And there's one more question always comes up that I hear is of palatability. Yeasts seem to be very palatable for animals, correct?
I think that is a general characterization of most yeast products. I won't say that that's going to be the case, across the board, but for the most part, we see at least neutral to positive benefits when it comes to palatability. And we have I'm going to say our postbiotic material, safmanin. That is a really, really nice postbiotic material. It's say the two functional ingredients are mannooligosaccharides, or MOS, which are really effective oligosaccharides in terms of binding pathogenic bacteria to sort of control populations. And then it's rich in beta glucans as well, which can be very promoting nutrients from an immune standpoint. So we get both. I'm going to say from the use of that postbiotic and material, it's going to, one, reduce down the pathogenic bacteria, but also improve local immunity function in the dog or the cat.
Gotcha. So thanks for that discussion, you guys. I think we've covered a lot of the questions that we received before the show, and we've been getting a lot more of them now. So I'm going to go through these, and we'll start taking some questions from the audience. But first, Russ, you had mentioned that you wanted to chat with Jan a little bit about what we've covered so far. If you guys wanted to go ahead and if there's anything that you wanted to discuss, I'll get these questions ready, and I'll be back in a sec.
Sure. Yeah. In terms of that, Jan, I was thinking back, in terms of when we first started. There was a lot of effort where I'm going to say we were looking at a single shift in a single strain of bacteria. And then we'd look, I'm going to say, looked at where we are right now. I mean, what do you feel like in terms of, going back, 25 years to where we were looking at those single strains, and then coming forward where we're now trying to look at these ecosystems. In terms of that, what do you feel like that we need to do historically to go back and make sense of some of those past studies that were really well done, but we just simply didn't know to look deep enough?
It's a great point. I think, yeah, we learned a lot. And I mean, I mentioned before the show in the little discussion that when I started with the microbiome, right, back then the expectation was that the probiotic is going to change the whole microbiome, right, or prebiotic is going to completely change the microbiome and so on. I think that was the wrong expectations. We just learned so much more about the complex of the microbiome, right, all those new bacteria and so on. And that also makes sense why the expectations that we had back then were wrong, right? Because back then you said, okay, this one probiotic is going to now improve diarrhea, improve digestibility and so on. And so I don't think there was nothing wrong with it. We just expanded. All the data is correct. I always say data never lies, right? It's just you just get new interpretation. And I think for me, even in GI disease now, we're pushing to this multimodal therapy approach. You have to address everything from different mechanisms at the same time. That's going to be the optimal, if that makes sense, optimal approach to maintaining health, right? So that includes diet, has to be highly digestible. Then you're adding prebiotics and maybe specific probiotics that we know really can prevent some of those clear damages that our animals gonna have over their lifetime, right? So I think it's just a natural kind of continuation of what we learned back then, and the data is still correct. It's just we expanding our knowledge about it now.
Right. Yeah, great build. Yeah, great build.
Yeah, thanks, guys. Let's see what some of the topics are on the minds of the pet food professionals who have joined us out there. So one question we had is if you could share how biotics support growth and recovery and body systems, and what specific biotics can offer these benefits. And also, what effects do biotics, especially yeast-based ones, have on palatability? I think we kind of touched on that a little bit before.
I'll let Russ answer that because he has done a lot of personal studies in this space. So
Yeah, and Tim, I'm just going to ask you to repeat the first part of that again to make sure that I got it.
Yeah, it was going into maybe other than GI health, looking at how biotics can support growth and recovery, perhaps after an illness or surgery, something like that, and other body systems. Basically, kind of like what are some of the other benefits of biotics?
Yeah, so I think in terms of there's not as much, I'm going to say there's not as much data available with regard to the growth aspect, but we know that probiotics in general are known to aid with digestion in terms of I'm going to say overall gastrointestinal health, nutrient digestion, thus increasing bioavailability. And I think any of those with regard to is going to be a positive with regard to growth. Now, certainly in the livestock segments, right? When we look at like piglet growth, this is one that's recognized and is trackable. They typically see improvements when in terms of like feed to gain ratios or growth rates. We don't typically take those type of measurements in puppies or kittens, but we would anticipate that a healthier GI system is going to be beneficial across the board when it comes to the performance of an animal.
Gotcha. So speaking of health and safety, the other question that we had here was, are there any safety or dosage considerations when considering or incorporating yeast-derived products into a pet food or a supplement? Can there be too much of a good thing, in other words?
Right. I think I mean, in terms of that, there's always going to be an upper limit as to if whether there's going to be an adverse effect. I'm going to say in terms of probiotics, we tend to as an industry we tend to target that around 1 billion organisms as an efficacious dose. But I think Jan knows in terms of some of his efforts, as he mentioned, there are trillions of bacteria within the gastrointestinal tract. So while a billion does seem like a large number, it is not a large shift in terms of the total population. And certainly there have been probiotic supplements that contained 12 to 15 billion per dose that has been used safely. So I think in terms of the likelihood of, I'm going to say, overdosing with a probiotic is going to be a low risk, but it's certainly possible.
So
Yeah,
going,
No, I mean, I think that's perfectly, absolutely correct. I mean, even with FMT, when we give a lot of bacteria orally and so on, we have very few side effects, right? But I think you always have to see if you give something orally, right? In a small intestine, there's not many bacteria. And so sometimes the adjustment periods, right? So you you can have a little bit gas, like, a little bit, So I think safety studies have to be done to ideal to find for the right probiotic, the right dose kind of, right? So those studies are important, I think.
so as we're going from talking about the health of the animals and the safety of the animals themselves, let's talk about the health of the planet. Are there sustainability aspects to yeast-derived biotics or other yeast-derived solutions for pet food in terms of the production or use of them as byproducts, things like that?
yeah. I mean, it's certainly in terms of one of our stance is the sustainability when it comes to the production of our products. Um, we Lasaf has been in the fermentation business for a long time, 170 plus years, involved in producing fermentation products. And with that, we have gained a lot of expertise over that 170 plus years with regards to producing microorganisms in the fermentation environment, as well as health solutions that are derived from the yeast or micro or other bacteria, microorganisms. So certainly from a sustainability standpoint, we feel like that we are being responsible in terms of the production of the products that we produce for the dog and the cat, and that they can be part of a sustainability program for any of the manufacturer's program.
Gotcha. So switching on to a kind of a more technical topic here, can alpha diversity of gut microbiota naturally fluctuate during specific life stages? For instance, in infants, we observe a potential reduction in diversity concurrent with an increase in bifidobacterium species, a phenomenon that is entirely physiological. This raises an important question. Is it potentially misleading to view increased alpha diversity as an unconditional marker of health?
Uh, I mean, I could talk about this topic for many for many hours because there's two separate questions here. One of them is a technical question: how do we analyze alpha diversity? Okay, and so alpha diversity is a very, very difficult statistical measure and depends on study to study has shown different alpha diversity measures and so on. So it's even a very, very difficult concept in my opinion. Um, but it clearly fluctuates, and there's plenty of data, there's studies out there that in puppies, right, when they're born, or even humans, we have a very low diverse microbiota because the gut is not yet developed, right? So the bacteria that come are all from the environment. We have a lot of E. coli, very few of the anaerobes. That is a very low diversity. And then within the first, very quickly, diversity increases. And then somehow the first 4 to 6 months of life, right, then where the whole microbiome kind of develops in response when the GI tract physiologically is mature in response to dietary changes, correct? So yes, so those are very strict. So alpha diversity itself has to be clearly put into a life stage, right? However, I also must say that even in clearly adult dogs, alpha diversity is all over the place. I see studies where people put them on new diets, alpha diversity goes down, and they still claim it's a good thing. And it's a very statistical concept. I'm not a big fan of alpha diversity in general. I'm kind of much more is which key bacteria should be there. And we know that in puppies, they're clearly different because puppies, the gut has to develop, be mature, functioning until it develops and so on.
Gotcha.
So life stage is very important, obviously, very young puppies. But then in the adult period is actually very stable. And then when they really get older, we see slight changes, probably because the GI tract doesn't function properly anymore.
Gotcha. So this is a question for Russ. What are the recommended inclusion levels of yeast-based solutions for optimal effectiveness in dogs and cats' diets? And is dry pet food better to include before extrusion or coating, or in dry pet food? Is it
Yeah,
better?
so in terms of it really is going to come down to the product that's being targeted. If we're talking about Safmanin or the postbiotic, our postbiotic material, uh, again, that can go into wet foods, that could go into dry foods. It's extruder stable. In terms of the matrix, it's going to dictate whether it's going to be, let's just say approximately 2% inclusion rate, two to three, depending upon the exact matrix of the diet that they are complementing. If we are talking the probiotic, our yeast probiotic, it is not recommended for wet foods. It's not going to be stable in that wet environment. It is sensitive to moisture. It's not going to be suitable to go through the extruder. It's going to have to be applied post extrusion. And with that, it's we have very, very good stability on the dry products. In terms of that, if we're talking, a semi-moist product, again, that's going to dramatically reduce down the shelf life of that probiotic as it absorbs that free water from the kibble and activates those cells. The way we obtain our stability is through the processing, we undergo a process called micropurling, where we take uh, dry yeast and we sort of sacrifice a small amount of yeast on the external coating of that micropurl, and then that micropurl can be then applied to the kibble. And those dead yeast on the outside, they sort of serve to buffer the amount of free water absorption, and that is to prevent the live yeast on the inside to being activated. Because once they get into a favorable environment, they are going to become metabolically active, and then we're going to be up against time with regard to how long they can remain alive, which is not very long once they become activated, unless they're in a favorable environment such as the GI tract.
Gotcha. So another kind of technical question here. How about the interaction among polyphenols and the gut microbiome? How does that happen, and is it beneficial to supply a yeast probiotic with polyphenols?
Now, you want to take the first stab at that, or?
I mean, generally, I don't think there's any reason not to combine different, functions that we know are beneficial. So I couldn't see, except the yeast would do something. But generally, I would don't believe there would be any disadvantage because again, polyphenols, especially the high molecular polyphenols, are converted by gut microbiota, right, into compounds that afterwards can be absorbed and so on. And so again, for me, if there's not clear mechanisms that we have tested, there should be no reason to exclude some of the different treatments at the same time.
Gotcha.
Yeah, I think in terms of that, I will just only add, when we start talking about, looking at overall nutrition, we don't, we don't just isolate that it's protein that's making a difference, or that it's fat that's making a difference, or it's the vitamins that are making a difference, or the minerals, right? It is that combination. It's a combination of those nutrients that are brought together that go into producing a complete and balanced diet. And we're starting to get more and more information where we can now start looking at this microbiome ecosystem as to what all are the influences, and which ones are going to make the biggest difference.
Gotcha. So another question here regarding the yeast interactions with other things. Are there beneficial responses to yeast beta-glucan strains? Or are the beneficial responses to yeast beta-glucans strain specific, or are the benefits ubiquitous for the species or species of genus or species of yeast?
Well, I mean, I think certainly from a standpoint that the beta glucan content is going to be important and it's going to be influenced by the specific strain of yeast that is utilized to generate it. Certainly, in terms of our SAF Mannan product, it contains greater than 20% SAF mannooligosaccharides, greater than 20% SAF glucans, and those are, we know that that's consistent from a standpoint of the strain that we use to generate that material. But I think in terms of that, I don't know the exact details of every strain of yeast, but there is going to be fluctuation with regard to one strain versus another. And certainly there are different forms of beta glucans, whether those are derived from other fungi or other sources. The exact composition of those beta glucans can be different and certainly have different physiological impacts.
Gotcha. So another question about the biology of biotics here. Is there any risk of gene transfer between probiotics and bacteria in the gut that could lead to antibiotic resistance? So is there any evidence either way, or is it just a myth?
I mean, theoretically for sure, right? But that's why probiotics have to be fully characterized before they can be used as probiotics. So that's why one of the criteria to fully characterize it understand the translocation properties and so on. Theoretically, yeah, but probiotics should be tested for that.
Gotcha. And so when we're talking about the yeast probiotics that Filio offers, would those yeast probiotics survive retorting for use in a wet pet food?
No, in terms of we do not recommend our probiotic for use in wet foods. One, I'm going to say the retort process would be pushing the envelope with regard to surviving, but the problem again is going to be the activation because of the moisture content. In terms of putting it into a wet product, the yeast are going to activate. And it's not going to be I'm going to say, a suitable environment for that to last long enough for an animal to consume it. So we do not, we recommend our product. We recommend for use in dry foods, dry treats.
Gotcha. And so we have a question here also about the products. Are they available in powder or liquid form, both? What's their format?
Yeah. So their Actisaf SC50 is the probiotic. It is a dry powder. And SAF Mannan is a dry powder.
And so it looks like we've gotten through most of our questions. I'll finish off with this last one here as we're getting close to the top of the hour. Are there any potential adverse effects, side effects, or any concerns when using a yeast-derived probiotic for a critically ill pet?
I mean, that's obviously a difficult question. It depends how you administer it, right? I mean, even with bacteria, but they're very rare. They're very, very rare case reports when, probiotics have been used in ICU settings, right? When you have catheters and so on, there could be potentially a risk for translocation. So in some extremely ill situations, I would be very careful, obviously. But generally, in most cases, at least there should be, there's no reported side effects.
Right.
Yeah, I think I would only add in terms of depending upon the type of illness, as John hinted, if it was severe trauma to the gastrointestinal tract, and there was a high, I'm going to say a higher increased risk of translocation, I think all veterinarians would probably approach with caution. We don't have very many published accounts of major issues, but I think anytime you use the term critically ill, it would, we would need to understand more about what that critically illness, condition was, and what where the trauma existed.
But just to give you a little perspective, I mean, there are studies have used probiotics in dogs with parvovirosis. There's also studies with fecal transplantation, right, in parvovirosis, and dogs with acute hemorrhagic diarrhea syndrome, which have a clear acute necrotic enteritis, and they turn to be safe. So again, I think in general, it's a very safe approach. but there's going to be a few patients, and so you need to look at the whole patient, right, and which disease it is that you want to be careful. But general, I would not be very concerned.
Gotcha.
Yeah.
All right. Well, great job. We managed to make it through all the questions, and we're only a minute over time. Did you guys have any final thoughts before we do close up?
Now, I just want to thank Jan for joining today, and look forward to hearing him this spring in Kansas City.
Thank you so much. Yeah, for me, it's just like I think that we need to, we just evolved the last few years, really understanding much more the complexity of the microbiome, how it interacts with the GI mucosa and so on. And I think this multimodal therapy approach, going away from I'm going to find one simple solution, but put this all in context of diet, prebiotics, probiotics, fiber, and then really selecting the best for which, type of it. So I think that is where really, I think we need to emphasize that.
Great. All right. So thanks again to Jan and Russ, and thank you to our international audience. We greatly appreciate your feedback on this chat today. So please scan the QR code that you should now be able to see, and we'll also email a survey link soon. I'd like to give a big thank you to Filio for sponsoring this edition of Ask the Pet Food Pro. And please watch for more Ask the Pet Food Pro chats brought to you by Pet Food Industry and Pet Food Forum. I'm Tim Wall, the senior reporter for Pet Food Industry, and I'd like to say thank you again to Filio, to Dr. Jan Sukodolski, head of microbiome sciences at the Gastrointestinal Laboratory at Texas A&M, and to Russ Kelly, and also a big thanks to the Watt Global Media staff for making this possible. And thank you to our international audience for joining us. I hope you all enjoy the rest of your day or evening, depending on which hemisphere you call home.
Yeah. Thank everyone for joining.
Thank you so much. Bye.